In this video, I will be showing you how to do an endospore staining. The materials that you will need for this experiment include malachite green stain, saffron stain, steam bath, paper towels, gloves, goggles, lab coat, a slide box, biohazardous waste container, stain collection vessel, germicide, heat fix slide, stain tray, slide clip, water bottle for rinsing, and bibulous paper. The purpose of the endospore stain technique is to determine if the bacterium on the slide is capable of producing spores. Spores are very resistant structures. They allow bacteria to survive in very harsh conditions. The first step in this experiment is to cover the heat fix slide with a piece of bibulous paper. The purpose of this paper is to hold the spore stain over the bacterial smear. Next, place the heat fix slide onto a slide rack that is over a steaming beaker of hot water or a steam bath. The steam bath is used to drive the spore stain into the spores, or bacterial cells. Then flood the smear with malachite green. This is your primary stain. Steam the slide for about 10 minutes. If the blotting paper looks like it's becoming dry, replenish it with malachite green as necessary. You need to keep the blotting paper moist. After 10 minutes, remove slide from steam bath using slide clip. Once the slide has been removed from the steam bath, remove the blotting paper. Then let the steam slide cool. After the slide has cooled, pick it up using the slide clip. Tilt the slide downward and rinse the smear in a gentle stream of water. The water acts as your decolorizer. Rinse until the excess malachite green is no more on the slide. Make sure to rinse the slide over your stain tray. Then place the slide back on the stain tray. Next, flood the smear with saffronin. You want to let this sit for 30 to 45 seconds. The saffronin acts as your counterstain. Then, rinse the excess saffronin off with water into the stain tray. Next, blot dry the slide in a packet of bibulous paper. Do this gently. Once the slide is dry, examine the specimen under the microscope using 1000x objective lens. This is what the specimen should look like under the microscope. The endospores will appear green, and the vegetative cells will appear reddish pink. After examining the specimen under the microscope, properly decontaminate yourself and your workspace. First, store your heat fix slide in a slide box for later use. Then, pour liquid from the stain tray into the stain collection vessel for disposal. Then, dispose of your gloves. Your gloves have been contaminated from the experiment. You want to put them in a biohazardous waste container. Then, wash your hands. Once you've done that, use germicide to wash down your workspace. After that, use paper towels to wipe up your workspace.